All right, we're rocking 2 screens today, so I'm going to keep doing this for the rest of the class. That should work. I'm going to try something slightly different, but hopefully this is big enough. Hopefully that works for everybody. So this is the first real content class of the day, or of the semester. This will be what I like to do with a lot of these lectures, and this will, especially this one. I'm kind of a wonk for science history as well, so I'll try to. I like to go back and kind of talk about some older stuff in order to introduce newer stuff and concepts, and especially with this class, it's kind of ripe for that because there's a lot of older microbial genetics that matters that builds upon what we're doing today. And so especially for today, we're going to talk about some older experiments. What I want you to get anytime I talk about experiments is not necessarily remember like, oh, this experiment happened in 1942. Like I'm not going to ask you what year it happened in or things like that. I want you, when we talk about these experiments, I want you to be able to understand what was going on. Understand that if I gave you data as a result of these experiments or as a reflective of what happened in these experiments, understand how to interpret that data. Understand if I change the rules of the world and say the world behaves this way, what will the data look like in that new world? So if you understand kind of what's going on in the experiment and the hypotheses and what's being tested, you should be able to understand how to interpret the data from those experiments. So just keep that in mind as I go over any experiments, and today I'm going to go over a few. So, but first I want to touch on what we're talking about in this class and what I'm going to go over, why it's important, and why I love what I do. So I'm a microbial geneticist. I'm a microbiologist. I am fascinated by this world. Microbes are everywhere, and they do a variety of different things. And so if you're just out in nature, if you just exist, so all of you guys, turn to the person left of you, to the right of you, you guys are like a cloud of microbes right now. So if you guys remember like Charlie Brown, I have no clue how old you are, but if you remember Charlie Brown, there was like a Pigpen character. You guys are like that right now. You just can't see it because you're constantly like shedding microbes and stuff. And you're actually, if we went in and measured it, you would start to reflect a little bit of the person sitting next to you's microbiome and likewise. And so you guys are all like a cloud of microbes, outside, inside. And basically we exist in a world where microbes are everywhere. And so you're starting to hear about this more and more every day as we talk about microbiomes and designing buildings based off of microbiomes and measuring microbiomes and how we might be able to manipulate those. But microbes are kind of everywhere in other aspects of that as well. So a lot of our bioengineering, a lot of our production for different food additives or products or medicines, drugs, microbes have a huge hand in that, and we're designing new ways every day to try to manipulate microbes for that purpose or for other closely related purposes. So up here on the left, that picture is actually a petri dish where the whole petri dish is covered with microbes. But what's happened is that these microbes are responsive to light, and when they see light, they drop down and die, or they drop down I think it's kind of a type of dye. So basically what has happened with this petri dish is that they've taken a screen in the shape of Einstein's head, shown that light on the petri dish, and wherever light showed, the microbes drop down, died. So you can actually use these to start painting and things like that as well. So, and you know, this may seem like, oh, it's just kind of why do this? Yeah, it's art, but also think of applications in the real world. So if we can have microbes that report what the environment is like, you know, there's people that are trying to design microbes that fluoresce and light up when they sense gunpowder or when they sense explosives. So anytime you fly, you know, they wipe your hands for the TSA. Basically what they're doing there is checking for different chemicals, for traces of bombs. People are trying to do that where they can just spray a harmless microbe on you and they light up if there's any trace of explosives. So there's a lot of microbial engineering things that are going on to design new generations of microbiotics. So top left is kind of cool. Top right, that is what the Earth looks like now and is only going to look like more and more in the future. So we throw out a ton of plastic into the world, a ton of plastic into the ocean. You guys are definitely old enough to remember the whole MH370 plane going down in the ocean thing. I don't know how well you followed it, but every day there was some report of like, oh, we found debris. Oh, it's just random plastic that was out in the ocean. There's a lot of garbage out there, and we really have no sense of this because you're obviously not out in the ocean, but there's a ton of garbage out there. There's a ton of garbage in landfills. We have oil spills everywhere, every day. There's an oil spill. The BP Horizon was a big one, but there's small ones every day. So one of the things that happened after the BP Horizon oil slick, basically one way to clean that up is to spray a bunch of microbes that eat oil. Oil is just, long chains of carbon. Microbes like to eat long chains of carbon, and so there's certain types of microbes that will actually eat up oil. Plastic's a little bit harder, but there's microbes that will eat plastics, even synthetic plastics like polyvinyl chloride or other, polystyrene a little bit. So one way we're trying to cure this plastic problem in the world is by engineering microbes that will actually break it down. If you look at plastic bottles, sometimes they say, this will be in a landfill for, they shame you into not using plastic because they say this will be in a landfill for thousands of years. Well, if we can design microbes that actually eat that plastic, potentially we can help take care of that problem. So we'll talk a little bit, actually the second discussion paper I think is actually on that topic, but we'll talk a little bit about that. Obviously, y'all are probably well informed about microbes in medicine. The next slide's going to hit that home, but microbes, they cause disease. They can help you out. There's beneficial microbes that are present inside your gut, on your skin, on your dog. There's beneficial microbes everywhere. There's harmful microbes everywhere. And so it really depends on the context how you look at that. But the reason there's on the bottom left here, there's a copper pot, is there's a lot of lore about how to control microbes, preventing us from getting sick. And so a lot of, I think the ancient Greeks used things like copper pots. I'm a microbe major. I'm not an ancient Greek major. Somebody way back when used copper pots. Why is copper pot good for storing water in? Well, if you put water into a copper pot, copper is one of the a reactive metal that will actually kill microbes on contact through a variety of ways, and there's not a lot of resistance to it. So even back when, if you just got water from a random, potentially contaminated by a pathogen source, if you put it into a copper pot, potentially those pathogens might have been killed. You might not have gotten salmonella or some other thing from it. So there's a lot of different treatments out there for microbes. We'll certainly talk about antibiotics and antibiotic resistance, but there's also some other ones out there where it's not necessarily intuitive how it works, but there's microbes matter for health and for safety and day in and day out. And then the bottom right is there's a lot of cool little things in nature that happen that really we don't know what to do with them yet, but they're in the future might be possible. Later in the class, I'll talk about CRISPRs. Those started in microbes. They're still in microbes, but they've been developed for genetic engineering and other things. The bottom right is some microbes. Some microbes, we're always taught in earlier classes that prokaryotes, no nucleus, no inner membrane organelles. That's wrong. Pretty much everything you guys are ever taught is wrong at some level, but in that regard, there's always exceptions, there's always outliers. So the bottom right here is actually a microbe that has organelles, really small organelles that can sense magnetic fields. So these organelles actually sense magnets. They can orient the microbial cells along the magnetic lines of the Earth. So yeah, it's just a cool system where there's genes involved in that, there's proteins involved in that. We can actually use microbial genetics to break down what's happening with pathways like that. And potentially you can imagine ways to engineer that or use that in different contexts going forward in the future. So there's a lot of diversity for microbes out there in terms of what they do and the pathways they encode. And there's a lot waiting to be figured out too. And so part of this class is giving you the tools to not only understand what's happening, but also think about potentially ways to figure out what the genes are involved in that microbial magnetic sensing, or how to engineer the pathway differently. So maybe it's only, maybe the dye is only expressed under UV light or under a certain wavelength of light or under a certain other chemical that's in the environment. So I'm going to try to give you the tools to at least start to think about that and design, understand how the cells and genomes work, and then design different ones for the future. But I wanted to just, the next slide is going to, this was released today. It was a CDC report today. On the news, the thing that gets the most press, obviously, is going to be things that kill humans. in whatever context we talk about them. Microbes have always done that, and they're going to start doing it a lot better than they've done it in a while. And so this was a report today out of Nevada. So for the last 60, 70 years or so, we've been pretty good at keeping a track of microbial infections and limiting the damage from pathogens in your bodies during surgery. You know, I just got kitties, so I have little cats at home, and they scratch the hell out of my arms. I have a lot of like small secondary infections because of that I don't worry about necessarily right now. But potentially in the future, you know, they go away, but maybe they don't. And if they don't, if they turn into a larger infection, thank God we have antibiotics now for that, but potentially in the future they might not. So there's a lot of things I think we take for granted in terms of the way we live right now that we take for granted because we haven't had to worry about microbes killing us because of stupid things that my cats do and whatnot. So this report came out today. It's got January 13, 2017 up there. It was a CDC report out of Nevada. So this is actually the first official government report in the US where they've identified a pathogenic isolate that is resistant to all known and all working antibiotics allowed to be used in the United States. So this bug is resistant. It's called Klebsiella pneumonia. It's resistant to, I think, 26 antibiotics they tried and that are used in the US. This is the first US isolate that's the woman. So the woman was 70 years old. A few years ago, she went to India, I think broke her leg there, had a secondary infection in the broken leg, was in a bunch of Indian hospitals, came back to Nevada, and then died of septicemia in Nevada. The thing that killed her was this Klebsiella that had gotten into her broken leg infection. And then we basically couldn't kill it because any antibiotic we had was useless. There was one antibiotic that still works, but it's not, it was not allowed to, it was not regulated to be used for bloodstream injections for this woman. So she died. This is you're gonna see a lot more stories like this in the US. This has been happening for a while, certainly in places like India and China, where there's a lot more incidents of multiple drug resistant infections. But we're going to start losing the ability to actually cure some of these microbes unless we figure out new ways to kill them. The other thing you'll see in the press, and it's in the press because, yeah, it gets clicks basically. So, like there's multi-drug resistant gonorrhea and syphilis coming back in the U.S. So people are going to start having that problem again too as well. It's not going to be you just go and get treated for random infections that you pick up. You're going to see this in the future be certainly in the near term. I think it's going to be a much bigger problem. We might be able to get the drug pipelines up going again and get more antibiotics regulated. But yeah, I think we're coming to the end of the world where we don't worry about some secondary infections of microbes in the U.S. And in other countries, they've been worried about this for a while. All right. Happy Friday. All right, so this is microbial genetics. We're going to talk about microbial genetics. We're going to talk about a variety of different things. The thing about microbial genetics, though, is that it's a pretty young science compared to other genetics. Immunology, Jenner figured, Jenner was smallpox vaccines. Not really vaccines, but kind of they took scabs off of cows and gave people vaccinations with them. So Jenner figured out a little bit about immunology, and really studies of immunology started back in the late 1700s. General genetics, Mendel started, he figured out some stuff mid-1800s, but really it was picked up about 50 years later. Virology started large scale in the early 1900s, actually as a way to treat bacterial infections. We'll talk about some of that later. There's actually a good book called The Microbe Hunters. It's a good book that was written in like 1910, so there's some anachronisms there that are not so great, but nonetheless, it was the second guy on there, De Herel, or I can never pronounce his name right, but it's the D''H''E', however you pronounce that. So he wrote a book called The Microbe Hunters. It's a great book if you're interested in kind of the behind-the-scenes science as it happened in the early 1900s. Again, with that warning, it's written from the early 1900s. But those guys were figuring out viruses back then and started the beginnings of virology, and they were working with what are called phage, which are bacterial viruses. Genetics really picked up in the early 1900s to 1910s, 1920s in T.H. Morgan's lab at Columbia. You guys probably heard of him with like fruit flies and genetics and things like that. So that picked up in the early 1900s and then continues today. Microbial genetics only started in about the 1940s though. And it started this time for a variety of reasons. So why do you guys think that people wouldn't give microbes a thought before like the 1940s? What would be holding it up compared to other things that have genetics? Yep. Exactly. They are. So the answer effectively is that microbes, when they knew, so Koch and Leeuwenhoek and earlier scientists knew that there were microbes, but really they looked very simple. And very simple physiologically, very simple morphologically compared to all these other systems that people were looking at. Especially in the 1800s, 1900s, where there was a lot of naturalists. You know, you had Darwin on a ship around the Pacific identifying all these cool organisms. You know, people were interested in going outside and identifying these fascinating organisms. And then you had a small group of people that are interested in microbes looking at globs on plates. And it wasn't the most charismatic of things. Because there was really no morphological complex diversity like in other organisms, there was a thought that potentially bacteria and prokaryotes didn't necessarily have genetics. Maybe they were there and they existed. So unlike the fancy fruit fly, maybe microbes didn't have genetics, and maybe we couldn't actually study that. So it took a while to overcome that. You'll learn, so when we talk about genetics, all of these genetic terms, genes, genetics, phenotype, all of these terms arose back before we knew what DNA was, before we knew what chromosomes really were. They arose as terms of convenience to identify and define something. So genes were the units of inheritance in fascinating organisms like fruit flies. But it was thought that maybe bacteria didn't have genes because they didn't have a lot of cool things to look at. So maybe there wasn't really a genetic basis for these things. They were just globs of goo on a petri dish. I will always say going forward, and I have this thought too, anytime you read these old papers, it's always hard to wrap my head around the old papers because we know so much more now compared to then. So you have to really get in the mindset of 1930s. All they could do is look at globs on plates, and that was bacteria. So it kind of makes sense that maybe they thought they were too simple to have genetics, and that they were just maybe bags of enzymes that functioned, but they couldn't do experiments with them or any, certainly no genetic experiments with them. So today I'm going to go over a couple of key experiments that really started to turn the tide, that gave people the feeling that, maybe there is something to this microbial genetics. Maybe there is something to studying microbes and trying to figure out what the genetic basis of traits are. So I'm going to go over two key experiments and then a couple of experiments that lead into those experiments. So the first key experiment I'm going to go over was actually published in 1944 from Avery, McLeod, and McCarty. Oswald Avery is that guy on the bottom left there, gigantic bald head. Avery was working on a microbe at the time called pneumococcus. It's now called Streptococcus pneumonia. The reason that Avery was working on that and that Griffiths before him was working on it, as I'll talk about in a second. So you guys know what happened in between 1914 and 1918? The big thing in the world happened. World War I, right? So a lot of people died. There was a lot of trench warfare. There was actually a lot of microbial pathogenesis. So in 1918 especially, there was a Spanish flu. Influenza came around, killed a quarter of the world. But the flu didn't actually kill most of those people. A lot of those people that died from the flu died from secondary bacterial infections. And one of the main secondary bacterial infections during World War I was caused by pneumococcus or Streptococcus pneumonia. So the pneumonia part of that is because it caused pneumonia. When your lungs are kind of filled with flu, and it causes other things to grow there as well. So there was a lot of research money from the Navy and from the Army and other places that was driven towards research into Streptococcus or pneumococcus. to figure out how to treat this, because if the Army cares about things when people can't fight, so we wanted to get those soldiers better so they can go and die in trenches later. But there's a lot of research money that was dedicated towards research into Streptococcus there. So the first couple of experiments worked with Streptococcus because of that exact reason. There was money there to do it. And so anytime I go over this, it's kind of, we'll talk about E. coli a little bit later. These early experiments were in a certain bug at the right time with the right place, where you can kind of imagine how it might have, what might have happened if the experiments happened in a different bug. Maybe things developed differently if Streptococcus wasn't killing people, if they had to work with E. coli. The experiments we'll talk about with Streptococcus, they wouldn't necessarily work in E. coli. And so there's things develop for a reason, and we figure out things in linear progression just by happenstance sometimes. And so the Avery McLeod-McCarty experiment in Streptococcus was what Avery was interested in, was that he wanted to follow up on a result that was published a decade or so earlier from Fred Griffiths' lab. And so what Griffiths was working on, and it was in Streptococcus as well, Griffiths knew that there were two different morphologies of Streptococcus on a plate. These morphologies were smooth, so big globular colonies on a plate, nice and smooth, compared to what was known as the rough morphology. So here on the left, they're smaller, but imagine that these are just tight little colonies that aren't covered in a bunch of goo. So rough, no goo; smooth, a lot of goo. There was a difference between these two colonies, that the rough colony, if you inject it into mice, so on the left here, the rough strain, it's non-virulent if you inject it into mice. And so if you take a syringe, stick it in a mouse doesn't die. The smooth strain, though, if you take a syringe, stick it in a mouse, the mouse dies, and it dies a pretty quick death. Turns over, closes its eyes, things like that. So we've got a rough strain, we've got a smooth strain. Not knowing anything else, what might you guys think about the difference from your immune system between the rough and the smooth strain? Yeah. Exactly. So the smooth strain is actually coated in, we'll talk about it later, but it's called EPS, or exopolysaccharides. This protects the cells, especially when there's a lot of them. It protects the cells from immune surveillance. And so one of the reasons that the strain on the right is actually virulent is because the immune system can't clear it, because it's hidden because of the smooth outer goo there. The rough strain doesn't have that, so the immune system clears it pretty quickly. All right, so that's the experiment going in. Griffiths went a little bit further, though, and the reason he did this, there's really no rhyme or reason. It's just, as scientists, sometimes you do things and they worked out. Really, there's no logic here to why he did this. There's another picture of rough and smooth strains. So what Griffiths did was he took so what you want to see when you have a virulent pathogen, you want to see, is that pathogen, living pathogen, is pathogenic growth necessary for virulence to happen? So what Griffiths did was that he took that smooth strain and then killed it. He boiled it, basically. He put it under a lot of heat. The cells died. So that all that's left, the EPS is left, the cell constituencies are left, everything chemically is the same between the heat-killed smooth strain and the smooth strain, except that the heat-killed smooth strains are dead. When you inject that into mice, the mouse lives. So we've got it's not essentially the bacteria presence itself, it's unique growing bacteria to actually cause virulence here. Again, the intuitive leap between this and the next experiment, nobody really knows. But then what Griffiths did, just to kind of do stuff in the lab, is he took that heat-killed smooth strain that can't kill mice, mixed it with the rough strain that can kill mice, injected that mixture into mice, and then the mouse went sleepy time on the bottom there. So the mouse dies. So whatever was in that heat-killed bacteria, it couldn't kill the mice itself, but if you mixed it with another non-virulent strain, the mouse could die. And so something in the killed S cells was transforming the R cells. And so because they used transformation in that definition, we call this phenomenon now transformation. And this was where Avery picked up on that. So Avery picked up, thinking that there was some transforming principle that could convert that non-virulent strain to a virulent strain. He wanted to figure out what that principle was. So So he wanted to, potentially there's a trait you can look at, it's virulence or not virulence. Potentially there's a genetic basis for this trait. Avery wanted to figure out what chemically was that genetic basis. And so he did. Avery was a really kind of fundamentally very detailed guy. And so he did what he usually would do, was he would take these dead cells and then try to piece apart and figure out what exactly was allowing transformation of that rough strain. And at the time, you know, the early 40s, what they could do was they could separate out, based on various chemical sieves, basically, they could separate out different constituencies within that heat-killed smooth strain to figure out what the transforming principle was. So the first thing he did, the first experiment that he did, he takes a culture of smooth cells, which could kill the mice, kill them by heat, stick them over a Bunsen burner that's not plugged in. That magically, in this picture, kills the cells by heat. And so then what you can do is extract that transforming principle and then put it into a variety of chemical tests. In this case, Avery is not looking at killing mice. He's actually looking at a difference in the morphology on plates. So he's a nice guy, didn't want to kill a lot of mice. So instead of looking at life or dead, he actually looked for the presence of smooth cells in the original plate that was rough cells. So he would mix the heat-killed S cells with a culture of R cells, plate them out, and look for these smooth, goopy colonies to show up. And then, yeah, he tried to figure out what the transforming principle was by putting in proteases. So proteases are going to be enzymes that degrade proteins. So if you put those into the culture of S cells, the mixture of S cells that's heat-killed, all the proteins are going to be destroyed, theoretically. So when you do that with proteases, you still get smooth cells showing up after the transformation. Same thing with RNases. So we knew what RNA was. We had enzymes that eliminated RNA. So you can put the RNase into the heat-killed smooth cells. You still got these smooth colonies showing up afterwards. And so there was the experimental progression here was just by the process of elimination. So using these two well-known techniques, he couldn't eliminate the transforming principle. The one that did eliminate the transforming principle, though, was, I'm going to have to figure out a better way to do this in the future. There we go. So the one that did eliminate the transforming principle, though, was if he took DNase. So DNase is going to take out DNA. If you put that into the S cell extract, it would eliminate all the DNA. If you mix that into the culture of R cells, you got no S cells back. You got no goopy cells back. And so this, in textbook form, nicely and clearly and definitively shows that DNA is the transforming principle. It actually took like a decade for people to actually believe this, because speaking as a person that writes papers, reviewers and critics are harsh on you sometimes. And so basically the critique of these experiments was that, well, you put in proteases, but you can never really get rid of all the proteins. Or you put in RNases, but you can't really be sure that all of the RNA is gone. So even though it's a clear experiment as we present it, it took a while for this to be believed. Yeah, so this is before Watson and Crick. This is before we knew what genetic was, what it meant. We knew that there were genes. There was some basis for inheritance for that trait. But really look at this as a chemistry experiment. So we knew that the transforming principle had proteins in it. They had RNA in it. They had DNA in it. DNA had been known had been known chemically for since the late 1800s. Same with RNA. So they're basically sugars, right? You can measure sugars. We knew that they were there. We just didn't know that that was a genetic material, or that it was what the structure of them was. So Watson and Crick showed the structure of DNA, but we didn't know that at this point. They didn't know that that was the structure or the genetic basis of anything. So they were just literally going. He was literally going through everything that they could measure and trying to eliminate one thing at a time to figure out what it was. All right. So at this point in the class, and this is typically how it's going to go, so we're about halfway through. When you guys see this slide come up, this is a signal for the online class that there's probably questions to answer that are relevant at this point. When you guys see this big star come up, it means that the next slide is going to be a question for you guys to answer. So when you see this, get ready with like top hat, however you're doing that. And then the question's going to show up. So think you're able to answer right now. Yeah, so yeah, as you log on, I can see the number. So you should be able to answer this. I'm going to ask some types of questions like this. This is one of the reasons I like Top Hat, but this is a way to introduce you to it. So on this slide is a question asking, what would the outcome be of the following experiment? So if you took the rough strain from Griffiths, you mixed in RNA from the smooth strain, and you injected it into mice, what would happen? So it's a pretty straightforward question. For the answer, what I want you to do is that there's a shaded box around the mice. You're just going to click on which one. Just one click. That'll answer it. This is the way I'm doing this very straightforward to introduce you to it. Later on, I can use this to do things like here's a structure of things. Where's the hydrogen bond that you're interested in, stuff like that. So I'll give you like another minute to figure things out. Last day to you can purchase Top Hat whenever. Yeah. Yeah. Oh. It's in the syllabus somewhere. It's 467499. And we'll give it like 20 more seconds. Yeah. Is it what? So if you're on the present, it should be showing you present. Are you in 2016? You're in 2016. That's why. Okay. Yeah. Yeah.
I'm normally in the online section, but it's not. But like I wanted, so I can't answer it now.
But you can answer it on the online one.
But I'm online.
So I haven't put that one up yet.
Oh, I have to wait.
Yeah. Yeah. All right. This was a demonstration time too, so if it's right or wrong, don't worry. I'm giving everybody credit for this one. Plus, if you log in later, just email me that you got it later. And this was just kind of to show you how I'm going to do it. And then, so everybody's going to get credit regardless. Plus, if you, this one time only, later if going forward, not going to work this way. It's one time only. If you do log in, just email me that you didn't have a subscription yet, you didn't log in, there was a problem, I'll just go back and give you credit for this guy. Yep. Just email me, it's fine. Yeah. It only works for this first class though. All right, so what I'm gonna do is close this. If I can. All right, I'm gonna close. Now you shouldn't be able to answer. And I can do a couple things with this. So I can show you the right answer, so it should show up as a bubble there. So if you take the rough strain is going to be non-virulent. If you put in RNA is not the transforming principle. DNA is the transforming principle. So in this case, if you put in RNA from the smooth strain, it's not going to transform the cells. The cells are still going to be non-virulent. Mouse is going to live on the bottom right there. The other thing I can do with this that I like is I can actually show you guys where everybody's clicking. So yeah, you really hammered that mouse lives. Good. But yeah, so this is, like I said, I'll do things like later on. I can put up structures of here's a piece, here's a structure of DNA. Point to the hydrogen bond that's at the 3' position, and I can do things like that, which I like versus just clickers. I'll mix in a bunch of different ones, but I like this for that reason. All right, so like I said, email me if there was any problem. I will, this one time only, I'll take care of it and then go back and retroactively make it so. Questions? Yeah.
Will we be able to see online, I guess, see our scores?
Yeah. Yep, that is the other nice part about this is that it automatically, it's in the cloud, so it automatically registers what your points are at any given moment. So, yeah, you won't be. So you'll be able to see it through Top Hat on D2L. I just have to go in and move them over. But the linking up, yeah, it's there's a lot behind the scenes that is not straightforward. Yep. Yep. If it was DNA, that would be the experiment that Griffiths originally did. Yep. Yeah, so right now, so that question is going to be what I'll do after today. I'll take that PDF. So that PDF for today is going to go up on D2L. That question is not, but there'll be it'll always be in Top Hat for review if you want to go in and review it. Yeah. All right. Make sense? All right. Second main experiment is going to be 1943. So this was published the year before Avery McLeod McCarty. It happened at this wonderful place that I grew up 5 minutes down the road from called Cold Spring Harbor. Cold Spring Harbor ends up being, it's really definitive, like, when you start to learn about the history of genetics, like a lot of stuff happened there. I had no clue until like 25 years after I grew up. But it's an amazing little lab that was a private little lab that a lot of early genetics experiments happened there. And so this experiment is going to be Lurie and Delbruck's fluctuation test. So the reason this happened in 1943, and this is, again, part of the confluence of events that led people to actually do these experiments. Lurie was a doctor, a medical doctor at the University of Indiana. Delbruck was actually a physicist. So Delbruck was a physicist that was coming over to biology. The timing is interesting here because what happened between 1939 and 1945? World War II, right? Turns out a lot of world wars have a huge motivation for microbial genetics, apparently. So what happened in the early 40s through the late 40s, you guys have probably heard of the Manhattan Project. There was a lot of physicists brought in, paid by the government to start to develop nuclear weapons. A lot of really smart people from places like Germany and otherwise were kind of fleeing to the US. A lot of these smart physicists were kind of scared by the prospects of nuclear weapons and whatnot. And so you ended up, there was a lot of physicists started moving into biology and asking more fundamental questions that they thought that they could use their mathematical frameworks for asking questions, maybe apply that to biology and learn some new things versus what had been done in the past. So Delbruck was a physicist that had a very mathematical mind. Lurie was a microbiologist, a doctor that wasn't very mathematical. But they got together and started talking at Cold Spring Harbor one summer and figured out that, well, maybe there's this cool experiment that we can do to answer a fundamental question. So the question that they wanted to address, though, was they knew mutations happened. At this point, we had a handle that mutations occurred. They occurred seemingly randomly in the world. We certainly knew that from T.H. Morgan's fly work because he had an army of undergrads kind of picking and looking for different flies. But there was two camps that were debating and arguing over the nature of mutations. So one camp thought that mutations just spontaneously occurred whenever. The other camp thought that potentially mutations only occurred after exposure to some stimulus. And in this case, a lot of people were, well, worrying about radiation from things like nuclear weapons. So two camps. Mutations occur spontaneously versus mutations only occur after some stimulus forces the cells to have mutations. Right. The scientific world knew that mutations happened. They didn't even know what mutations really were, though. They knew that mutations changed physical traits, but they had no clue, like, again, 1943, didn't know DNA was a genetic material, didn't know, had no handle for a lot of the basis of what traits were, but they knew mutations happened. So what Lurie and Delbruck wanted to do was quantify the state of mutations and then design an experiment to test, which one of these camps is right. Do mutations occur randomly, or do they occur only after exposure? And so they used this statistical approach, mainly driven by Delbruck's mathematical way of thinking, to look at E. coli. So they were growing E. coli, and they were looking at E. coli resistance to viruses, or phage. So they had a line of phage that would kill E. coli. If you plated out the phage and E. coli on a petri dish, there would no E. coli would grow. But every once in a while, you would get a resistant colony that was resistant to being killed by the phage that you mixed it with. So they used this trait as a way to test. They knew that whenever they got resistance to the phage, that was some type of mutation, and they set up an experiment to test when those mutations actually happened. It was all based on, it was not direct. It was not a direct measurement of mutations per se. The experiment was based on comparing statistical distributions and thinking logically about what had to happen to give the outcomes there. So the way that they set up the experiment, it's pretty straightforward. So you can grow up E. coli in two different ways. The key here was that no matter which way you were growing it up, whether it be in one large culture or in many small cultures, if you had a large culture that was 20 mils of liquid and you had 20 small cultures that were 1 mil of liquid, basically that was you were growing up 20 mils of bacteria. It was just how they were sampled that differs between those two. And so the reason that becomes important is because you can use that difference to ask the question of when mutations occur and are they random or not. So if you assume that both the large culture and the many small cultures have the same amount of total cells in them, so whether it be 20 mils versus 20 times 1 mil, that's going to be the same amount of cells. So if you played out a 1 mil sample from the culture on the left, so if you played out a 1 mil sample from this culture on the left on one Petri dish, and you do that 20 times, that's going to be the same sampling-wise as if you took 20 cultures that were 1 mil and plated them out each one time. So if you do that, the difference between how those colonies grow up allows you to test this idea that when mutations occur. So if all cells have an equal chance of becoming resistant upon exposure to phage, so if all cells only become resistant after the phage is added, it doesn't matter if you grow up 1 mil in 20 cultures or 20 mils in one culture. You're plating the same amount of bacteria on each plate. And so if you're only putting the phage on the plate after the bacteria are there, it's the same chance of mutations happening if mutations only happen after exposure. Yeah. Yep, so I'm going to get to that in one slide, but yep, you were ahead of me. Key take home from this, though, same amount of cells on each plate. If resistance only occurs after the phage are added, doesn't matter how you grow it up, it's the same amount of cells on each Petri dish. However, so this is basically a showing of that. So if you played out 8 dishes, and there's 8 cells total with 1/8 chance of becoming resistant, and each cell, on the plate, if there's a 1/8 chance of becoming resistant, each plate is going to have a 1/8 chance of becoming resistant. That's what that shows. So yeah, no matter which plate, 1 out of 8. You can think about, if it was 16 cells, it might be if it's a 1/8 chance of becoming resistant and there's 16 cells, how many are resistant? 2, right? If there's 24, 3. So if there's only 1/8 chance of becoming resistant after exposure, you get the same amount of cells resistant on each plate. So this contrasts with the other worldview. So if mutations occur spontaneously, they're going to be occurring no matter when the cells are growing. They're going to be occurring when they grow in a large culture. They're going to be occurring when they grow in small cultures. They're going to be occurring before you plate them out on the plate, before you expose them to phage. And so in this case, the way that they thought about this was that it was actually Luria was at these happened back then. So Luria at the University of Indiana was at a casino night. They actually had the university-sponsored casino night. And he was at a slot machine, and he was just playing slots and then figured out, sometimes you get nothing back, but sometimes you get a jackpot back. And so that gambling led to Luria to this thinking about it this way. So if mutations occur spontaneously and they're constantly occurring during cell growth, there's a few different distributions that might have depending on when mutations occur. So if you're growing a culture of cells and there's a random mutation that happens late in the culture's growth, there's not going to be many colonies on the plate that are resistant, because if you start with 1 cell and let it grow to, in this case, 16 cells, or 8 cells, it's 5 o'clock at night, so my brain doesn't work great. If only 1 mutation occurs, and that mutation occurs late after division, so we've had 3 divisions here, you get 1 resistant colony on the plate. Only 1 mutation has occurred, it occurs late, you get 1 colony on the plate. However, if that mutation occurs in the very first division, still only 1 mutation happens during this culture. If it happens early, you're going to get a much different distribution on the plate, even though that mutation, it's only 1 mutation happening. And the reason for this is that if it happens early, a random early event for a mutation, that mutation is going to happen, and then that cell is going to divide again and again and again. And so you get many progeny from that initially resistant cell versus the first example where the mutation only occurred, in one cell late in growth. So this changes, as opposed to that mutations only occur after exposure example. Only one mutation occurs here, but depending on when it occurs, it's going to give you different distributions of resistant mutants on the plate. And so on and so forth. And so if you got intermediate events, you might get 2 mutants, 4 mutants. Same, only one mutation is happening, but it's happening at different points in cell division during that culture. So you get different numbers on the plates afterwards. So the whole basis of the Luria-Delbruck experiment was to grow up cells in these 2 different conditions, one main culture versus a lot of smaller little cultures, and then plate them out on plates, plate the exact same number of cells out on plates, just grown up differentially, and then measure the number of mutants that happen after exposure to phage. And remember, if it's one worldview where mutants only happen after exposure to phage, you're going to get the same number of cells on the plate regardless of which way you grow it up. However, if mutations are occurring as things are growing and before phage, during the growth, depending on when that mutation occurs with the second worldview, you're going to get a much different distribution of cells that are resistant on those plates. So in this case, this is basically how the experiment, I think these are numbers from the experiment. So you've got one large culture, you're going to plate that out 20 times, and there's only, there may be 1 or 2 or 3, 4 mutants that happen in that one large culture. But as you're plating out that whole cell, there's only a certain amount of resistant mutants within that one large culture. And so when you plate it out on a bunch of plates, you're basically just sampling the average and then the spread around the average for however many mutants are in that one culture. However, if, with this on the right here, when you plate out many smaller cultures, each of these cultures, there might be a different mutant that happens at a different time within these cultures. So if you plate out one culture, maybe a random late event happens. Another culture, maybe a random early event. Another culture, maybe no mutation happens. And so when you plate out one sample per culture and they've grown up independently, you might get a very different distribution than the ones that just from one single culture. And so when I talk about this experiment, I'm talking about indirect measurement and indirect measurement of mutants as the hypothesis, because they're not really measuring when mutations happen. They're measuring the statistical distributions that happen based off of how they set up the experiments. And this will become a thing in the next lecture. Because again, this is one of those experiments that a lot of biologists weren't really math savvy, so they didn't necessarily believe this experiment, even though the numbers were pretty clear. When Luria and Delbruck actually set up the experiment, you get the same means, so that it's the same average number of mutants basically, depending on how you grow them up, but the variance between plates is much different when you grow them up in a variety of little cultures. So this difference in variance showed them that it had to be that mutations were occurring randomly during growth, before exposure, because there was this, there were mutants within the cells and within the cultures before they actually plated them out on the plate. But again, it was indirect evidence, and so people didn't necessarily believe it for a while. Actually, I can get through this last part. In the last 2 minutes, I can do this. So people didn't really believe it until there was direct evidence that mutations were happening. Direct evidence. One of the experiments that showed this, and the other one that we'll talk about, is in the first discussion paper. But the first direct evidence that mutations occurred spontaneously just during the growth was published in this paper by Josh Lederberg and Esther Lederberg. Esther Lederberg was Josh's wife at the time, gets much less credit than she should, but she was kind of a superstar of early microbiology as well. She gets kind of dwarfed by her Nobel Prize winning husband, but shouldn't. Nonetheless, this experiment showed that there was direct measure, they were able to directly measure when mutations were happening, and so they can directly test that mutations were happening spontaneously over time. The way they did this was by growing up E. coli on a plate and then looking for antibiotic resistance. When you grow up E. coli, if you just grow it up in a lawn on a plate, it's going to grow up as a lawn. What you can do is take that lawn of bacteria and replica plate them to another culture, to another antibiotic plates. The next slide is going to be a YouTube link to replica plating if you don't understand what that is. But when you take this culture and lawn, if you replica plate that lawn of bacteria and then put an antibiotic plate on top of that velvet for the replica plating of the culture, what you're going to see is that mutant colonies show up. This plate on the right here is basically, think of that, that's an ampicillin plate that's got ampicillin in it that's been replica plated from that lawn on the left. What you see is that colonies show up, antibiotic resistant colonies show up in certain parts of the plate. You can do this multiple times, though, so you can replicate that same starter culture to different, and there's the YouTube video. You can replicate that same starter culture to different plates. And the reason this is a direct measurement of when mutations happen was because if mutations only happen after exposure, the mutations aren't going to happen until they hit that replica plate, until they hit the ampicillin plate. And so if mutations only happen after exposure, you would expect that mutations happen in different places on successive replica plates. But since mutations happen spontaneously, it means that the mutants they're already present within that lawn. They're already present on that first plate. And so when you replica plate, there's going to be some slop to it, but the mutant cells, the mutant colonies, are going to be in the exact same places as they were on that original plate. And so this was a direct measurement of when mutants happen, kind of opened the doors to really people truly believing that microbes had genetics. Bunch of other stuff happened after that we'll talk about. One last thing to mention, you guys don't have class Monday, so if you show up, I'm not going to be here. Have a good MLK day. At the end of each lecture, or I'm going to try at the end of each lecture, to have take-home points. So think of this as a summary. People always ask me, oh, can we have a review sheet? I'm giving you this at the end of each lecture. If I were to make a review sheet, I would basically just put this down on that. So take that into mind as well. So these summaries, things, if I'm mentioning it here, I probably want you to understand what those things are. I'll see you guys Wednesday.